Good morning, Dr. Amit Dutt. I'm Dr. Poonam here. Good morning.
Oh, hello. Good morning. How are you?
Yeah, so good morning, everyone. I welcome Dr. Amit Dutt for today's class, which is genetic characterization for the tongue cancers, the various he will be talking on this topic, and so you can start.
Sure. I'm just sharing my slide. Let me know if you can see the slides. Can you see the slides?
Yes, we can.
Perfect. Okay. So good morning, everyone. Good morning, Poonam and Deeta. Thank you for having me. So I hope I'm audible enough, right? You guys can hear me.
Yes, you are audible, sir.
Perfect. Perfect. So today I'm going to talk about, I would start with some basics of next generation sequencing platform and its application. And then what I would do is that I would go into that how we are using this next generation sequencing platform in deriving biological sense and then how can this be turned back onto the clinics. So basically questions which are coming from the clinics to the laboratory, and then how we would see that it can go back onto the clinics. I could hear some background. Yep. So so my laboratory primarily at ACTRAC has been working on different cancers, mainly the head and neck cancer, lung cancer, and also on the breast cancer. But today I'm going to be talking about is the work of a graduate student from the lab, Bhaskar Dharawat, and along with all the work done with Dr. Sudhir Nair, a collaborator, our long-standing collaborator right from the beginning. More or less, we had a similar time that we joined at ACTRAC together. So the work, all the work that I will be talking about today is in collaboration with Dr. Sudhir Nair. So, but before, as I said earlier, before I start into the research problem and to the research question, I would want to introduce some basics about the approach that what we take, that is using the next generation sequencing platform. So let's start from the basics. Just one slide of basics, and then we will move on to the next generation sequencing. So first of all, let us define the problem. What exactly is the problem? The problem is to, as what we all agree over here, that cancer is a genetic disease. So what really underlies this genetic disease are the different kind of like insults to the genome. So if you would want to find problems, find solutions, you would have to first define the problem. So the problems are the different kind of insults to the genome. So let's catalog them in a very simplified manner. What are the different kind of like insults? So one of the insults could be, as what you are all aware of, there could be a point mutation in the genome, in the DNA. And these point mutations can lead to change in the amino acid, and that may lead to either an activation of a gene or an inactivation of a gene of a class called as tumor suppressor gene. In addition to the point mutations, there are structural alterations that we get to see. What are these structural alterations? We get to see that there are change in copy number. Either there could be an additional copy number, as what you are seeing, which is called, very frequently called as, also referred to as amplification. And this is something which is an hallmark of oncogenes which are resident in this region. and the similar side, what you would see over here in the blue band is a region which is deleted, so that is lost, or there could be a loss of heterozygosity. So this could be a hallmark, a change in the copy number of a gene called as the tumor suppressor gene. In addition to the oncogenes, in addition to the copy number alterations like amplification and deletion, there are other different kind of like alterations, such as the translocation, in which gene from chromosome A gets physically transformed and equal to some other chromosome, and its expression is regulated down there. So in addition to this, there is another mode of infection, another mode of insult that what we kind of like get to see more often now. And now that with the power of the next generation sequencing, we are able to find more of these. We are finding more and more of non-human elements associated with the genome, that is the infectious agents. In addition, now we also know that there is another level, higher order of alteration, which is called as an epigenetic alteration, where the DNA does not harbor any mutation, but the modification onto the DNA, or how the DNA is compact in the chromosome, that modification is altered. That is the epigenetic modification. So these are the major insults. Now, if you would want to do it, and how people have been doing it as of now, you might be aware of. If you want to figure out the point mutation, then the good old way, the standard way has been, is to do a direct sequencing. That is a Sanger sequencing, correct? If you want to figure out if there is a copy number alteration, such as an amplification or a deletion, you would want to figure out by earlier there was a comparative genomic hybridization, that is CGH. Then it moved on to microarrays. Then it moved on to SNP arrays. So basically, those microarray based platforms would tell you the copy number alterations. But these sequencing, the Sanger sequencing would not tell you about the copy number alteration. Similarly, the microarray platform will not. Similarly, the microarray platform will not tell you about the underlying point mutations. If you would want to know for these translocation kind of an event, neither of these, the microarray or the sequencing, will help you with that. So for that, you will have to perform, which is routinely done in the laboratory, is like the fish kind of like an experiment to identify different regions of chromosomes. Similarly, if you want to do in a conventional way of an infectious agent which is present in the genome, what is classically required is an a priori information. You need to know what infectious agent you are looking for. For example, if you are wanting to see HPV, you need to know the sequence of the HPV. Then you will design the primers, and then you will amplify the HPV in that way. And similarly, there are modified way of sequencing, of Sanger sequencing, where you chemically treat the DNA and perform, and that will allow you to tell you what different kind of like modifications are there. So what this is telling you is that the conventional or the classical technologies are distinct for different insults, for the different alterations that underlie the genome, and that makes, and then also they are very much underpowered. I'll show you in another slide that what are the major limitations of those, and that's why we require a completely a change in the way that people had been approaching, and leading to the complete a disruptive platform such as the next generation sequencing. So here is a very clinical problem that what we kind of like routinely get to see. So as what you know, that most of the tumor samples, they wouldn't come out like a very pure tumor sample. There would always be a stromal contamination, a normal DNA contamination, right? But then what is also that what we are seeing usually in tumor samples, that there is an inherent problem of aneuploidy, or inherent problem of, as what I kind of like said, like copy number alterations. So here is a typical example of mutation in a gene that has only 1 of 5 copies. You know that if you would be looking at this gene in the blood, that is in the germline, you will be finding only 2 copies. And the genotype would be A, AA, AB, or BB. However, here what you're seeing is that there is a mutation in a gene which has now 5 copies in total, and of these 5 copies, only one of the allele is mutated. Okay, so basically there is a 20% allele which is mutated at that loci. Now, if you further confound the problem and then you make it that 40% of that of that sample is tumor, rest is normal, the actual allele fraction will come down from 20% over here to 8% because there is only 40% tumor, rest is all normal, where it would be deployed. So let's see how the classical conventional technologies have been addressing this, or if they can address this. Here is the classical SICAM, because this is a mutation that you want to figure out. Here is an experiment that is being performed, which we call as an admixture experiment. What they do is like they take a cell line, and in that cell line, here is an heterozygous mutation. So in an ideal scenario, there is both the alleles are present in 50-50% frequency. However, now what you start doing is that you start reducing the tumor content, or you start increasing the normal content into that. So when the allele fraction drops at 40%, this is how you see the two different peaks. Here is the mutant peak, and here is the wild type peak. Still, you could kind of like have some kind of guesswork that there is something MS over here at this loci, and there could be an heterozygous mutation. However, if the tumor content comes down to 20%, you see, when the allele fraction comes down to 20%, you see that there is very difficult to make a distinction between the background and the mutation, which kind of like exists around over there. And here we are talking about 8% mutant frequency, and this is a typical sample. We always tend to get to see this. So with the conventional technologies, that is with the Sanger sequencing, it would be very close to impossible. I'm using the word impossible to identify those mutations which are occurring at a lower frequency. So that's why that's not the technology that we could kind of like use for doing a discovery experiment. For a validation where we already know what mutations are, that could still be twisted and tweaked around. Similarly, not just at the sequencing, if you would see at the copy number alteration, as what I said, copy number alterations are performed by microarrays or SNP arrays, and that are primarily for amplification or deletion. Here, the copy number alteration is being used to infer loss of heterozygosity. And what we are seeing is that here is again a pure cell line. And if you would see, the loss of heterozygosity is shown in blue color. So if you take a pure cell line, you see that it is all blue. When you start reducing the content of the tumor with adding a admixture of normal DNA into that cell line, what you tend to see is that if the normal content, if the tumor content is below 60%, that is, if the normal content exceeds 40%, which we always invariably most of the times that you would kind of like get to see, unless you really have very high quality samples, you would see that it's all yellow. That means the blue is lost. That means there would be no way you would be able to figure out whether this region harbors and loss of heterozygosity or if it does not. So what is this telling us? That the different technologies such as the Sanger sequencing or the SNP arrays, they have their own limitation in resolution to, and especially when it comes down to the cancer settings, it becomes difficult to use these technologies to make novel discoveries. If it is for germline genetics, it would still be kind of like, okay, if you're looking for Mendelian or those kind of like disorders, but not in the cancer, in the settings of cancer, where the genome is totally at flux and there is like lots of different nuances that underlies in the genome. So what do we do? Classically, what people have been doing, I've taken this slide from internet. For a moment, you can kind of like just ignore this guy. So what this guy is doing is that he is in the classical way that people have been approaching, is like sequencing doing a piecemeal approach, doing sequencing of one gene, and then the second, oh, this gene also does not have a mutation. Sequence the third gene, oh, this gene, but that is impossible. In fact, it took 13 long years to sequence the human genome at a cost of like more than $1 billion. And today, the same thing can be done even at ACTRAC at a cost of about $1,000. That is about ₹1 lakh actually. And this can be done around within less than a week. So you can imagine the change in the difference in the technologies that it is, the disruptive technologies has brought in. So this guy is wanting to look for a signal, which is right over here. And if it takes a piecemeal approach, you can imagine how much time and effort that it would take for him. The chances of him to land up into a true signal would be more than by defined by the matter of chance. What does the genomics do? This is what we do within the genomics and using the next generation sequencing platform. So here in the genomics, we just illuminate everything, and what you can kind of like see. So there are a lot of other additional data as well that what you kind of like get to see, but then you are able to identify your true signal using these genomic approaches in a much more. So that's why you would require a lot of statistical approaches as well, where the true signal has to be distinct from the noise that it kind of like generates with the high throughput data set. And this is how a typical next generation sequencing platform data looks like. This is a cartoon which has been taken from this review article, and this shows that the data which comes out from a next generation sequencing. I'm not introducing you today to the chemistry of next generation sequencing platform. I'm only kind of like telling you the limitation of the conventional platform and the need for a disruptive technology. And here comes the next generation sequencing. So what happens in the next generation sequencing? First of all, that the reads are small. Okay, so these are the small reads which are coming out of a platform. And because we are in a reseq era, that is, the human genome has already been sequenced, right? So every cancer sample that what we are sequencing, we already know the reference of it. That is the template, so which is kind of shown over here. So what happens is that when you take the small reads, you have to computationally, you have to mathematically align it back to the reference sequence. And once you do that alignment, what information do you get? You get that as what you can see over here, that there is a allele A at this chromosome loci in the reference genome. However, most of the reads have C, so which is suggestive of that there is a point mutation in this region. What we are seeing over here is that there is a gap, there is a deletion over here, across in most of the reads, which is suggestive of a small indel or a small deletion in this region. What is happening here that none of the reads aligns to this region in the chromosome, human chromosome. So that means, what does that mean? That means there was no read, from this region, suggesting that there is a homozygous deletion likely in that in that sample that what we have taken. If you would take the median read, what is shown over here is that the median read is half the read of the whole genome that what you get to see in these samples. So there is an hemizygous deletion. There is only one chromosome of the two. Here, what we are seeing is that copy number amplification, because the number of reads in this region exceeds the number. And here, what we are seeing is something very funky happening, is that half of the read is aligning to chromosome 1 and half of the reads are aligning to chromosome 5. What does that mean? That there is a potential translocation event that has happened over here. And lo and behold, what we are also finding in these kind of like high throughput next generation sequencing data set is that there are set of regions, set of sequences that does not align to the human genome at all. What could that be? Then what you can do is that you can align. My laboratory is a big time into looking for pathogens, and along with Sudhir and I, we have kind of like make some very interesting discoveries in novel pathogens which are associated in oral cancer. I won't be talking about that today, but essentially what the way it is done is that in any NGS output, you take the reads which do not align to the human genome, and then you align it and into the microbial genome and see if it is any of the known pathogens. So what is this slide telling you? As I said earlier, you require distinct and different technologies for the different alterations. NGS is a one-stop solution. In this one experiment, you are finding mutation, you're finding delete copy number alteration, or the different kinds of copy number alteration, and you're also finding if there is any pathogens. And similarly, even the epigenetic alterations could also be identified in just one experiment. So that's the power, and that's the disruptive nature of the next generation sequencing platform. That's what, so that was a kind of a brief introduction. Also, I want to kind of like now say now that because we have talked about next generation sequencing, there are different kinds of next generation sequencing. You may have heard about, very frequently you would be hearing about people asking for whole genome sequencing, or whole exome sequencing, or targeted resequencing, or whole transcriptome sequencing. What are those, and when do you perform those? So it all depends on what question that you are asking. For example, in whole genome sequencing, human genome has 3 billion bases. So to sequence all of those 3 billion bases is something which is not clinically relevant, because it's possible that you might find different mutations. And with this 3 billion bases, you will find that there will be a lot of noise out over there. And to find something which is statistically significant, and at the same time that there is an approved treatment for it, the chances are very low, because those treatments which are already approved, you have a defined set of it. So absolutely, in clinical settings, whole genome sequencing for the cancer setting is not a great option. In whole exome sequencing, what happens, about 1 to 2% of the human genome of the 3 billion bases, around 50 megabase, is the region which actually codes the coding region, the genes. There are about 20,000 odd genes in the human genome. Depending on what you call as gene and what, so because now there are non-coding genes as well, and that you would also club as a gene. So that some people would say it is 25,000 genes, some people would say 20,000 genes, but both of them are correct, because those who are saying 25,000 genes, they are including the non-coding genes as well. So if you are sequencing those guys, just the coding region, which is kind of like more relevant, then you are performing a whole exome sequencing. And similarly, if you are wanting to sequence whole transcriptome sequencing, that all the genes which are converted into mRNA, and then that is called as either an mRNA sequencing or whole transcriptome sequencing, to understand if there is only amplification at the DNA level, or if there is a concomitant overexpression also at the RNA level. Something which is of the most relevant into the clinics is the targeted gene sequencing. As what I said, let's take a step back. There is only X amount of FDA approved treatment, X number of FDA approved treatment. Then why sequence genes for which there is no treatment? Yes, you can make a discovery, but then that is not clinically relevant. So what people do, they make smaller panels. They don't even want to sequence 20,000 genes. If you would want to look for genes which are actually FDA approved and for which there is a treatment, they are kind of, you can count on your fingers, it's less than 20. So then why sequence 20,000 genes? So for that, what people do, including the germline and other different, it is about like 50 odd. So people do, depending on the need and depending on the tumor type, they would design different gene panels. They would define different gene panels, and these gene panels for the number of genes that might be relevant for your disease, and that is more relevant, which is more cost effective. Because you're sequencing smaller genome size, and also if you find some alterations, there would be a treatment out over there. So these are the different applications of next generation sequencing, which has already gone into the clinics, where a targeted gene sequencing is something which is very frequently being used. So now let's come on to the research question, that using this NGS, that what have my laboratory along with Dr. Sudhir Nair's laboratory, what have we been doing together all the while that we have been here at ACTRAC? We joined, as what I said earlier, we joined ACTRAC around on the similar time frame. So here is a classical paper that you all are very well aware of, by Dr. Anil G. Cruz. And the question over here was that in the N0 trial, that those guys who are clinically node negative, in those folks, if you would compare the therapeutic arm with the elective surgery, you find that elective surgery is associated with a better overall survival. However, as what you are aware of, this elective surgery also comes along at a cost, at some major comorbidities associated with it. And there has a stalking number who can be recused, who need not go into the surgery, and that is about like 70-odd%. So having some kind of a biomarker is the question that both Dr. Sudhir Nair and myself, we were very interested in asking if we could compare these samples with the node negative and the node positive design experiments and look for biomarkers that underlies and that can tell us that could tell us that to meet the underlying unmet need. Identify which of the samples among these N0s, which of the patients in these N0 stage are likely to metastasize and known. So what did we do for that? This is one of the first studies with about 58 odd sample, but then now we have done much more studies. We have, I wouldn't be giving you the complete whole, the recent studies with around 295 samples, but I wouldn't be talking about in details about like all of those studies. So in the first study, that what we took here is the classical table one, that what we kind of like call in any of these studies, the characteristics of different samples. They are all early stage tumor, T1, T2, and we are keeping ourselves as homogeneous as possible, is that they are all tongue. So my laboratory is also very much interested in developing computational tools because we deal around with such high throughput data sets. So one of the tools that what we developed initially is, we call it as an Hpv detector that was published in the Journal of Cancer, much and very well cited paper, and that allows people to figure out whether, based on the NGS platform, whether Hpv is present there or not. So there has been a lot of like noise and different groups talking about whether the tongue cancer are associated with HPV or not in the Indian settings. We found that of all the 58 sample, and then now we have an extended study as well, none of them are that we found are HPV associated at all. They are all HPV negative, and this was confirmed by our HPV detector and the other standard assays. So this is how the sample of ours kind of like looked for. What did we do? We did not do the whole genome sequencing. Yes, we have done whole genome sequencing of these samples, but that is not the focus of the talk today. What I'm going to talk about is that what were the leads that what we found from an whole exome sequencing. That is, we were sequencing only the coding regions, which is about 50 megabases of the whole genome in these samples. So, and what is shown over here is a heat map representation. That's a standard way how you represent these mutations. Each column is a patient, and each row is a gene, and the blocks, the solid blocks, represents mutation in that gene in that patient. Okay, so the first question that what you would do in any of these kind of like studies, because these were one of the first early studies actually from India in tongue cancer, there hasn't been this at this resolution that had been explored. So the first question that you would want to ask is that, okay, there is a set of mutations which is already known in TCGA or other different kind of like studies in tongue cancer and other different oral cancers. So the first question you would want to ask, are we totally different? Are we really going to be, I mean, do we harbor the Indian patients a similar mutation profile, at least in the hallmark genes that you would expect it to be? So this is what it is kind of like showing over here. Here are the data sets from the Caucasian, and this is the frequency of mutation of these genes. And what we find is that most of these hallmark genes are altered at a comparable frequency as what you would otherwise expect from these studies, which was kind of like great. But then, and at the same time also, this is showing the copy number alteration. This is showing an amplification. On the left is like deletions. What is shown is primarily an amplification. So the genes that you would expect to be amplified, that what we found in our studies were consistent. What came out from this surprise is that in next few slides, I'm going to take you through some biology, and I'm going to take you through that how a lead from an unbiased lead from a genomic study is taken into the laboratory, and then how it can be turned back into the clinics. So here I'm going to talk about a gene which is a Notch1. Okay, so Notch1 is a receptor, and what was initially seen, that what we found is that the mutation in the Notch1 was about 20% in our sample, as you see in this study, and which is kind of comparable as reported in literature in the TCGA. So it shouldn't have been like that eye-bruising at all. But what was reported in literature around at that time is that Notch1 in TCGA is a tumor suppressor gene. What does it should be inactivated, it should be deleted, and it should have a stop codon kind of like things in the Notch1. However, the graduate student in the laboratory, Pawan Upadhyay, what he found is that majority of the mutations, what we found in our samples in tongue, is that none of them were inactivating mutation. So we were asking question, are we missing out inactivating mutation? Are we missing out? And in fact, what we found that some of them mutations looked activating in nature. Now that was kind of like paradoxical. You would not expect a tumor suppressor gene to harbor an activation, because that's a hallmark of an oncogene. And not only this, even when we looked at the copy number alteration, we found that the notch was amplified to a certain extent, which was statistically significant. Now, none of these were getting consistent with the literature. So again, going back onto the basics, Sudhir and I, we are deliberating about it, that we really do not understand what is happening in here. Could this be an oncogene? So let's do it and let's see it. Most of the work that I'm talking about here now, it's already kind of like published as what is kind of like shown over here. But the reason I'm still kind of like talking about is because I want to give you a flavor how these leads lead to allow you to design experiments in the laboratory, how you validate it, and then how you turn it back onto the clinics. So that was the mutation profile heat map representation that I just showed you. Now I've changed slides, and here in this, what is a validation experiment. So what Pawan is doing over here is that he's taking experiments, he's taking those samples, and then he's validating the next generation, the leads from the next generation platform by using orthologous technologies. So what is here? Here is at the DNA copy number. He performs a real-time PCR and to look at the copy number alterations. And what he finds that it is not just the Notch1, but what he does is that he takes all the members of the Notch signaling pathway. Okay, so if you would take different members of the signaling pathway, and then if you would ask, are those all altered or they're not altered, or it's only the alteration is specific to the Notch1? What he finds is that there was a significant number of patients which are harboring, and now that was a kind of like a very interesting lead, very interesting finding. We are finding that yes, indeed, there were samples. In blue, it is shown as a deletion. Indeed, there are samples which is consistent with the literature. Literature says that Notch1 is a tumor suppressor, so it should be deleted. Yes, we do find patients which are deleted where it might be a tumor suppressor, but at the same time, we found samples which are not only mutated, but they are also amplified, and not just Notch1, all the components of the pathway. Not just at the DNA level, when we look at the RNA, again, we find a similar trend. The samples which are harboring amplification at the DNA are also, which doesn't have to be all the time. We can talk about it. Also led to an overexpression of the transcript. So you can see that there was a good statistical correlation in the sample where it was amplified and where it was overexpressed. Now, this was again kind of like suggestive that maybe there is something distinct happening in these sample sets. And to remind you, you are very well aware of that these samples of Indian origin are having a distinct etiological agent associated with them, right? Which is different from what you normally get to see in the Caucasian population, and primarily it's the tobacco chewing. So we started asking a question is that this could be something related, associated with tobacco. One of the first questions that what we asked, which I'm not talking about today, is that there are distinct signatures. I'm not taking you into the details of what exactly is a signature of the mutation, that we count the number of mutation, how many times it is A to T, how many times it is T to C, and then there is a mathematical way to calculate a signature. And then each of these agents, etiological agents, they have their own defined distinct signature. So yes, we found that there was an enrichment of the signature, which was associated with tobacco. So which was all consistent with what we were kind of like finding. But then in clinics, what you would find, if this is something really true at the genomic level, then can we really kind of like see it in the IHC? So yes, when we performed the IHC, we could find that there were about 40% of like these samples that were significantly overexpressed in these samples. So now the question was, are these different subclass of tongue cancer? So what we do is, like, one of the first questions that you would ask, the literature says that Notch1 is a tumor suppressor. You are finding an overexpression. So the question is that can we show it with the cells in the laboratory that these are activating in nature? Yes, we can design an experiment to address those. How do we do that? What we do is, like, we take cell lines which are derived from Indian origin patients. Here there is, that was this cell lines were not established by my laboratory, but that was established in 1984, Tata K et al. At Tata Memorial Center, and we got as a legacy those cell lines. So if what you normally do is that here what you're seeing is a Western representation, that is the proteins in these. And when you look for the protein in this cell line, you find that there is a good expression. So what you essentially do is that you use a technology called as an shRNA, that is a knockdown. You obliterate, you ablate the genetic expression of that gene by using an shRNA molecule. And what you're seeing over here is an shRNA 1, 2, 3. And what you can see, even though the same amount of protein was loaded across, here there was a reduction in the protein of this cell line of Notch. So that means now the cell line which was expressing X amount of Notch1, now we have reduce the expression. And when we reduce the expression in this cell line, we want to see what are the phenotypes that are associated with it. And what did we find? That the transforming ability of this cell was significantly reduced. This was not true just in one cell line, but in 3 different cell lines. So and not just the transforming ability. We were interested to understand if there is a metastatic readout, which is the migration of the cell. And we found that even that was distinctly, that was shown in the previous slide, that was also distinctly reduced. But why are we so excited about that Notch1? We are excited about this Notch1 because there are approved treatments against Notch1. And there are pharmacological inhibitors that inhibits the Notch1. So here what you're seeing is including the inhibitor, that was a gamma secretase inhibitor. That's what we use. Whenever we add the gamma secretase inhibitor, we see that the activation of the Notch is gone across the cell line, is reduced. And so the genetic ablation by knocking down the gene, or doing a pharmacological ablation, that is by inhibiting the protein, we find a similar response in the cell-based studies. And what you can see are that the cells, they tend to get like sensitive to the treatment of these. So let's see what it means clinically. So there were other properties as well, such as the stem cell forming properties. Even those were, all you need to know is that even those were significantly reduced when we use the inhibitors. Clinically, when we are wanting to see if it is associated with any of the phenotypes, and what you do is that you take the different genes in the pathway. Here are the target genes which are downstream, and here is the Notch, which is up.
कॉर्नर नहीं इधर हाँ एक्सट्रीम कॉर्नर ऐसे नहीं ऐसे बिल्कुल कॉर्नर ऐसे नॉर्मल हम स्वाइप करते हैं ना वैसे हाँ कैमरा देखो कैमरा तो दूर चलो हाँ
Oh, hello. Can you hear me?
Yes, sir.
Oh, I'm terribly sorry. I mean, this is the other side of like the problems that I mean, my system just froze. I didn't realize. I kept on presenting. I kept on talking, and when the slide didn't move, I kind of like. So, can you see my slide back again?
Yes, sir. We can see the slide, sir.
Okay. So, I mean, I have lost time about like maybe 10 minutes or so. So, but how does your schedule? Is it okay if I extend it for 10 more minutes, or should I
No problem.
stop at 10:30?
Sir, you can extend, sir. No problem, sir.
That's okay
I
with your
don't
time.
think
Okay.
we'll-
Okay.
Yeah, no problem.
Okay,
Yeah.
thanks, and sorry for that. So yes, so what I was talking about, was another gene that is the MMP10. I'll run through this. I will not kind of like take into the details. I'm only kind of like I'm only wanting to tell you is that given that we were wanting to look for biomarkers which were predictive, and Notch was one of the candidate. The another candidate gene was an MMP10, and that we found to be significantly higher in the tumor. So, but then again, this was a lead from a genomic study, and we want to now. I will take you through now through the next few slides. Again, this would be a biology again. That what you do in a research laboratory, how you address this question, and then we will see that how it goes back onto the clinic. So this is a work of another graduate student in the lab, that is Bhaskar Dharawat, who is submitting soon. What Bhaskar Dharawat does is that, okay, so now you have found from the genomic studies that there is a gene MMP10 that is statistically significantly overexpressed in those folks where there is a lymph node metastasis, and found to be not so high in those where there is no lymph node metastasis. So what does he do is that he takes a cell line, again an oral cancer cell line. This is actually a tongue cancer cell line established from Indian patient. And what you do is that you overexpress. You take this gene MMP10 and then you overexpress in the cell. And what you're seeing over here is a significant overexpression. Here there is a control cell, and here you see a significant overexpression. And when you do a significant overexpression, that is a forced overexpression, you see that the cell acquires certain phenotypes. And what are those phenotypes that is being looked over here? This is an assay which looks for a migration. And what you're seeing over here is that the cells are migrating more. There is an assay which looks for wound closure, and it closes the wound. So we make a wound onto the cell, and then it has to close. So we find that when you overexpress MMP10, there is a higher migration. Not only higher migration, when we do a specific assay for invasion, we find that the cells are also having a higher invasive property. Interesting. These are all consistent with what you would have otherwise expected. So then what he does is that, you know, this shows the sufficiency that when you overexpress, it is sufficient to have a higher migration potential. But then is it also essential? That is another question. For that, what you do is that you take another set of cell line. Here we are taking an AW8507 cell line. And what you're doing is that you are knocking down the gene. So you see that here in this cell line, so this cell line has a lower expression endogenous. This cell line AW8507, it is also a tongue cancer cell line, has a basal expression which is significant. And here it starts knocking down by an shRNA. And when you knock down, reduce the expression of this protein, what you see that there is a decrease in the phenotype, that is the migration and the invasion. Now this is both consistent. So it is not just sufficient when you overexpress, but then it is also essential, such that if you would remove the gene, the cells are not able to migrate or do an invade. And this is something that he goes on to show with other cell line as well, to show that this is not just a cell line specific event, but that would be something that would be associated with this gene in most of these tongue cancer cell lines. So that was an in vitro experiment. That's how you plan to kind of like to summarize in this way. But then you want to now bring it to a mouse model system and in vivo settings, and you want to kind of like see whether whether this is kind of like really true in the in vivo settings as well. So what we do is like we make an orthotopic mouse model. What is an orthotopic mouse model in this case? It's a mouse, it's a tongue orthotopic mouse model. So what you do is that you take these cell lines, and then you have already seen that you're genetically modified it. You are knocking down the gene, you're overexpressing the gene. Now you take these cell lines, and then you directly inject, you pull out, you anesthetize the mice, and you directly inject these cells into the tongue of these guys. So as what I will kind of like show you over here. So when you inject these as an orthotopic injection, now this requires a bit of a dexterity. I mean, for us, it is kind of like a big thing. As what you see, there is going to be a bulge over here, the cells as it get injected, and then this liquid kind of like it recedes over the time. And then you keep monitoring these mice, and then we now know that exactly about like in 25 or 26 days, what we get to see in these mice is something as follows. We start seeing the tumor formation. So that means that we have established an orthotopic mouse model, tongue cancer mouse model in the laboratory. Now that makes it amenable to ask several different kind of like questions that otherwise you wouldn't be able to ask in human. Here's what you kind of like get to see. This is something about like on 25 to 28 days old, post injection. So, and these cells, what we do is like they are also luciferase tagged. So you can kind of like do some kind of like an imaging, and what you're seeing over here in these cells, that these are the tumor formation. And the interesting part that what we kind of like get to see is that here is a cell line where the endogenous expression of the MMP2 is knocked down, is decreased with an shRNA, and what you see is that it is still forming the tumors in both of these cell line and in cell lines. However, when you are this is a negative control, and this is the actual knockdown of the gene. When you knock down the gene, we see that there is a reduction in the tumor formation, as what you see over here. And not just a reduction in the tumor formation, the more important question is what happens to the metastasis. So you are injecting it here into the tongue, and what you get to see is that primarily the metastasis we see in the lymph nodes, but then at times we have also seen metastasis into other organs. This was a rare, but we do get to see. And when you knock down the gene, what you get to see is shown in this panel. So when you take 6 mice, and when you take negative control, that is, you take shRNA, but which is not specific to your gene, but a generic one, you see that all of them are forming metastasis to the lymph node or to the organs. And when you knock down the gene, the metastasis is all gone, which is kind of like suggestive that this gene is essential. Also in vivo settings, these evidences suggest it is essential and involved in metastasis. What about the clinical setting? So there comes our good old study, that the N0 trial. Sudhir and I, we kind of like contacted Dr. G. Cruz, and we said that, okay, can we get like those samples from the N0 trial? And if we can kind of validate our finding as a biomarker in that study. And Dr. G. Cruz was very, very generous. He allowed us the permission to use that. And then comes in board, our very close collaborator, Dr. Munita Bal. We work very closely with her. Many thanks to her for trusting in our work and contributing significantly. And at the same time, Dr. Poonam Gera at ACTRAC, every day the students will be running down to her and seeking her help in these. So with their help, what we do is that we perform an IHC, we perform with the MMP10, and what we find is shown over here. First, let us look at what happens to at the level of transcripts. So just the RNA, we perform in real-time PCR and look for whether the expression of RNA is higher in the node positive or not. And we find that in these samples from the N0, yes, the RNA expression was higher in those folks where there was a node positivity. And not only at the level of RNA, when we look at the protein level as well, we find that there was a significant association of MMP10 expression with the with the nodal positivity. So which kind of like makes us that okay. So this tells us that okay, so there is something as a biomarker which will qualify as a biomarker, and this study is yet to be communicated, and there is more that what we have kind of like done onto this study. In another slide, I would kind of like show you that we don't just restrict ourselves to the MMP10. We have dissected, this is primarily the work of Bhaskar. Bhaskar has dissected the whole pathway. He asked the question, what regulates the expression of MMP10 in these nodal positive cells? So what he does is there's a lot of like mechanistic work, and he finds microRNA. Cutting a long story short, how this was discovered is not important. What he finds is that MIR944, one of the microRNA, directly, and this is biochemical experiment using luciferase tags and all. He shows that this microRNA directly binds to the 3' UTR of MMP10 and downregulates its expression. And when we do the genetic perturbations in cells, that is, we overexpress the microRNA or downregulate the MMP10, they both have a similar phenotype. So basically what it is trying to do is that in these experiments, we are dissecting the pathway, and it also kind of like finds a very interesting. So now you have a lot of reagents in the lab. You have cells which is overexpressing MMP10. You have cells which have knocked down with the MMP10. You have cells with different constructs and different cell lines. What you can do is like you can take NGS next generation sequencing again, and you can perform an whole transcriptome sequencing, and you can see a differential expression in these, and you can try and make. That's what he did. What he finds again is an interesting discovery, that is when the MMP10 is overexpressed or down or it's knocked down, we find that there was a significant association of another pathway, that is an AXL, the AXL signaling pathway. Now, AXL is an interesting signaling pathway, given that we know that there is already an FDA-approved treatment to it, that is the crizotinib, which is approved against the ALK, ROS, and AXL signaling pathway. I wouldn't take you into the details, but the reason I still have this busy slide for you is only to tell you that in the laboratory, what you do is like from the leads from this, you dissect the whole pathway. You find what is upstream to MM. First, you find that MMP10 is associated with metastasis. Then you find what regulates MMP10, and then what is downstream to MMP10. And then that gives you the power, an additional power is that, and then you ask question if all of them are having a similar phenotype, because they are in a linear, they're in a pathway, and that's what you kind of like go ahead and add rigor to your analysis, to your study, and that what is kind of like really been done. And one of the most relevant slide it shows is that the IHC from all the studies that what we have done from our own N0 studies and also from the TCGA, we find that the expression of MMP10 is very significantly associated with the nodal positivity. This is true not just in our study, it is true in the TCGA studies as well. And based on the transcriptome data as well, that they are very significantly associated. So where are we heading to? We are heading to make a discovery over here. This study should soon be communicated, where we are identified a biomarker which we are hopeful that it would also stand the time or stand the rigor of the clinics. We really do not know. These are mostly yet to be validated into the clinics. But the good thing is, only yesterday, I was having a good word with Dr. Sudhir. He is very excited about it. Now that's something that is the strength of ACTRAC, where we have basic scientists and the clinicians working together, and each one of them are excited with each other. So now what happens is, like, if Sudhir is excited, that would lead to a clinical trial, a clinical validation. Otherwise, majority of the times, these kind of like leads, they never get to see onto the clinics, and they never kind of like you're not able to test it in the clinics. But now with the collaboration and with the intense close work that what we do, this would be kind of like, so that's the story that I'm kind of, there are several other kind of like leads I did not talk about. We have found some novel fusion elements. We have found some novel fusion transcripts which are associated in head and neck cancer. Then we are found some novel pathogens. I've not talked about like those, but these are all long collaborative studies. And I've only talked about, we started with the notch as a biomarker, and I introduced you MMP10 as a biomarker, which very likely would be going. So these are the students from my laboratory who have been working around. And of course, we are very grateful for our to our collaborator, with a specific mention of Kavita from Dr. Sudhir Nair's group. Exceptional help that what we have received from her, from handling of the samples, without all which this wouldn't have been possible. And of course, all our collaborators. This also, these kind of studies, they require a lot of funds. It has been very generously funded by the Tata Memorial Center, by the Department of Biotechnology, by the Wellcome Trust India Alliance, NCI, DST, Department of Science and Technology, and the Terry Fox Foundation. So with that, I bring an end to my talk. I'm happy to take questions, should there be some. And again, apology for the intermittent disruption. Thank you.
Sir, thank you so much. It was an excellent class, as expected, and you made really a relatively complex topic a simpler one for us to understand as clinicians. I think, sir, if there are any questions, we'll be taking them. Till then, I would just ask one or two questions. One is that, sir, is there any difference in mutation between these tongue cancers and our routine buccal mucosa and gingivobuccal cancers? Because most of the time we see that these the P53 mutations, the notch, and the other group of these, whatever I have read, is they are almost similar. So how it makes it different? I mean to say that these mutations are almost, I think they are similar or they are different. Yeah, I'm talking about Indian patients only.
Right. So a quick answer to your question is that end of the day, they are all squamous, right? So the majority of the mutation profiling that what we have seen, we have performed, for example, for tongue. Then there is an ICGC, Indian Cancer Genomics Consortium, that was earlier led by Dr. Rajiv Sareen and Dr. Partha Majumdar. They have done for buccal mucosa. Then there are other groups who have also done it for tongue. And when we look at the overlap, actually do not have that slide over here, you find that majority of the players are overlapping. They are kind of common. In fact, to an extent, we have specifically looked at the MMP10 and the notch, because we were primarily interested in them, and we find that it is also upregulated in these tumors, because at least the notch we find to be like upregulated is making it's more of a difference between a tumor and a normal, and that tumor could be any subtype in the oral. That's what we are seeing. For MMP10, it is slightly different, where we are seeing more of an association with the metastasis. So we are primarily restricting ourselves only to the tongue. A short answer. A straight answer to your question is that majority of the mutations, being the squamous, are comparable. In fact, if you would compare the lung squamous or even from cervical cancer, the hallmarks of the squamous is something that you get to see across, and that would you get to see in here as well. But then there are distinct in the site specific. Cervical cancer has some other major players, lung squamous have some other major players which would be absent in oral, but within that oral, the other different subtypes, they all appear to have a common mutation profile.
Right, sir. So, but if I would like to add it from the clinical perspective, so this has been our experience, and moreover, few studies have also shown that in fact, the advanced head and neck cancers, if we compare them between the tongue cancers and the buccal mucosa, gingivobuccal sulcus, usually the tongue behave more aggressively.
Yes.
So even though the genetic profile and the other things, they look similar, but still probably there is some difference, and probably in future we will be looking into that also.
Definitely. I've never said they're same. Yeah. Yes, they are similar, they're comparable, but there is more to it. And a short answer to your question is that we don't completely understand. At this point of time, we are still in a cataloging phase, okay? We are cataloging the different alterations, but what alterations is actually causal to the phenotype that what we get to see, we have a very sparse understanding. We are very much early on. We are at an infancy for that. So we are, yes, what you said is absolutely correct. What I'm telling you is with regards to the genomic alterations, that is comparable, but then we know that there might be something much more that we still need to kind of like tap on, and we have not yet reached there to assign genetic correlates to all the phenotypes that what we get to see in the clinics.
Correct, sir. So another question, I think it's a simple straightforward, especially for the residents, that even though the EGFR is expressed in around more than 90% of the head neck cancers, and this is a routine question actually, and they are over in them, And so why still the clinical response is not very good, sir? Because whatever the trial we have or the data we have, even for the cetuximab, we have whatever data, it is basically for the metastatic or recurrent setting tumors. That one is the Bonner trial, the other is the extreme trial. And the mostly in the curative setting, if we even for the these good prognostic cancers like the HPV, the by Hisham Mihana, which is the de-escalate trial. Yes. So that also has not shown any benefit of cetuximab. Even it was found to be inferior to the cisplatin when those patients were treated. So what I mean to say is, even though they are expressed so much in head and neck cancers, so ideally they should respond very well to these therapies which we have presently, but we don't get the response like that. So what is your take on that, sir?
So it is very contextual, and it's very histological. I mean, site specific as well. For example, if you are coming from lung cancer, and in lung cancer, we get to see that there is a very significant response to EGFR mutations, and in breast cancer, you see amplifications of HER2. But then the EGFR, the same gene in the head and neck cancer, do not show us in that. But you have to also take into consideration the context of it. And the context is that head and neck cancer, there are several other different kind of like alterations. There, I mean, there is a very high incidence of P53 mutation and several other mutations that kind of co-occur with the EGFR. So EGFR itself is not a single player that you tend to get to see in lung cancer, for example. So maybe with more understanding of the underlying biology of like what are the correlated alterations that is present in the head and neck cancer that goes along with the EGFR, perhaps we might be able to do some kind of like combinatorial way along with the cetuximab, and that may make it more effective. As of now, you're correct that EGFR amplification itself is not just map treatment, or what are the other alterations which are responsible for it. We as of now do not have very clear understanding, but what we do know that they are it is not very surprising that it is a response that we do not really expect it to be, is because the cancer genome is totally nuanced. There are numerous other different kind of like alterations that happen in a very context what organ. Why we do not have a molecular, but then it's only like further research and over the time be able to find. I would imagine that there would be some kind of like combinatorial treatment along with the EGFR, maybe cetuximab, that would kind of like make it more more responsive to the treatment. But as of now, we really do not understand. I mean, there were trials done by Surya as well. I mean, the celecoxib along with the EGFR, and so there are several of those. Have a very clear answer.
Right, sir. I think, like you said, we need to have looked associated. I think the down is considered as one of the important, that there is some changes in that pathway which we are not taking care of, and the combination of these therapies would probably help. So one,
yes.
And the last question.
I mean, accessibility of the inhibitor, accessibility. I mean, in different. So there are several. I mean, there are several aspects to it that might kind of like get into. Yes, sorry, you were saying the last question.
No, sir. Sir, the last question from my side is, sir, that like we know, sir, that this is a common question. Even our patients, they often ask us that though my relative or the other person has been eating the tobacco for last 20 years, he has not developed cancer. But I have been eating it for 5 years, but I developed this cancer. So can you please explain to me that these are the common questions which we get from the patients or their relatives very often. So, sir, what is your take on that? That how does it happen, and what is the role of genetics into that?
Right, genetics play a very strong role, right, and a very strong play into this. So I mean, there is something as what we know, the germline predisposition, which even though has been shown only about like in 5 to 10% of like familial kind of like cancer. But then we know in majority of, and now there is more and more incidents, more and more evidences which are being cited of germline mutations which co-occur with somatic alterations. And we know that these germline alterations, I will take your argument rather to be in a slightly in a different setting as well. For example, EGFR alterations that we get to see in the lung cancer. There was recently a very classical study kind of like that was done, which looked for whether that it is the lineage of an individual of a patient that were exactly, because we know that it varies with ethnicity. And they were asking a question that people who are from Latin America, if they go on to for generations to the North America and they settle down over there, even though they have a different baseline EGFR mutation frequency, does the mutation frequency change with the habits of because now the environmental habits are environmental aspects are distinct. And what they find is like very surprising, or perhaps not so surprising, is that that it is the germline genetics which plays a much significant role. That means, for example, among Indians, the EGFR in lung cancer and adenocarcinomas are found to be about like 20-odd%, 21, 22. In Caucasian, it is 8%. So if Indians go and settle down in the US, Indian origin people settle down in the US, and even for generations, there have now the studies kind of like show that it is a germline genetics which plays a much stronger, their incidence of developing EGFR mutation in adenocarcinomas, lung adenocarcinomas would still remain as 21, 23%, and will not change to 80 to the Caucasian 8%, or to the Japanese more than 35%. So bringing back on to the question that what you are asking is that with these understanding of the significant role that the germline genetics play, possibly plays a much significant role in discerning between who are more likely to be developing cancer when they are exposed to tobacco, and those who are perhaps not so likely to develop. What are these germline predisposing genes or alterations? That is a subject of research again. A lot of studies have been done, but people have not been able to kind of, because there are several nuances in that, like the data. But over the time, I believe that this would be something that people would be able to address. The short answer to your question is it's in the germline, not just the smoking of the tobacco. Smoking of the tobacco is just the tip of the thing. If you're already having a predisposition, usage of tobacco or smoking of tobacco would kind of like make you more liable to develop cancer.
Right, sir. So, sir, I think this was a great class and we really learned a lot. I think there are not many questions. I think there is one have one class on aspects of these cancers. I think some this broad topic they have asked you to take some next time, and I would request you on behalf of our residents and students. So next time, if you we would like to again have you in our Saturday this class with another interesting topic. I think the topic they have written is one somebody has requested. I will just read it. The topic they have asked is, let us see, molecular methods in cancer. So some topic you want.
Molecular methods in cancer.
Methods in cancer, yeah.
So would that mean molecular methods means advanced molecular methods such as next generation sequencing, or the conventional molecular methods, real-time PCR,
I think we
standard
can,
sequencing?
I think, sir, we can do on both of them if you would be willing to, as per your convenience. So not every time, it will be as per your convenience. You can take on both the things, because I think it's important for us to know the conventional as well as the newer techniques, That will be
So,
the
so
better
Poonam,
one. They have written
I
send
must
their
say,
sequence and
I really love the enthusiasm. I mean, I like the enthusiasm. I mean, I'm
Definitely.
glad that you're asking these questions. I'm always happy to take any of these kind of like sessions. I'm happy to do that.
Oh,
And
thank you so
I'm glad
much,
to see that people are wanting and willing to. So, yep.
Yeah, thank you so much, sir. So we conclude
Sure.
today's class.
Okay.
Thank you so much.
Okay, thank you very much, and have a great day. And thank you again, Poonam and Tita, for having me to speaking today. Okay, bye.
Yes.